Welcome everyone, and thanks for joining. I'm Brenda Dettinger from BioPAC, and I will be your moderator on today's webinar, a new bioimpedance technique indexing dynamic activity of the sympathetic nervous system. Today's webinar is being recorded, and we'll send each of you a link once the recording is processed. During the webinar, if you have questions, please send them. Please submit them through our GoToWebinar control panel. That's how you can communicate with me, your moderator, and I'll pass questions on to the presenters as often as possible. Before we get to our presenters, I have an announcement. We host a biennial human physiology training event, and we've released the 2025 dates. We'll be hosting this event again in July of 2025. It will be at UC Santa Barbara in California, and we'd love to have you join us. It's a great opportunity to get hands-on experience with BioPAC devices, including the devices on today's webinar. Here are a few of my favorite photos from the 2023 event. Save the date, July 13th through the 17th, 2025, and meet us in California. Now, on to our presenters. We have an amazing panel of speakers today. We have Dr. Neil Dundon. He is an associate project scientist at UC Santa Barbara. He is a computational neuroscientist who studies how human physiology is associated with human decision making, with a specific emphasis on decisions to perform skillful motor actions. Welcome, Neil. We also have Elizabeth Reiser. She's graduated from the University of South Carolina and is now a PhD candidate in neuroscience at UC Santa Barbara. There, she utilizes advanced neuroimaging techniques to map brain connectivity in populations with neurological conditions and across the menstrual cycle. She also serves as the Psychophysiology Fellow for the Department of Psychology and Brain Sciences. Welcome, Liz.
Thank you.
We have Fraser Finlay, the CEO of BioPAC. He has over 25 years of experience in the life sciences and physiology monitoring industry. He is a well-regarded expert who presents at workshops all over the world. Welcome, Fraser.
Thank you, Brenda. Good morning, everyone.
And last but not least, we have Dr. Victoria Babenko. She received her PhD in cognitive neuroscience from UC Santa Barbara, where she researched the effects of naturally fluctuating sex hormones throughout the menstrual cycle. Victoria currently supports BioPAC customers and assists researchers in expanding their psychophysiological capabilities. Welcome, Victoria.
Hi, everyone. Thanks, Brenda.
Now, Victoria will review for us how stress researchers study the sympathetic nervous system. She will also introduce our new TREV methodology to do so and present some of her own research. Victoria, I'm making you presenter. Here we go.
Okay, so stress is traditionally thought of as a variety of mechanisms that the body undergoes in response to external and internal threats to homeostasis. And it has numerous direct as well as indirect effects on the brain and the body, from structural and functional plasticity of the brain to actions on the mitochondria and metabolism. And it can affect all sorts of aspects, including daily mood, decision-making, and bodily functions, as many of us are very aware. And for this reason, many researchers are interested in studying the effects of stress, primarily by measuring the behavior of the autonomic nervous system, and specifically its sympathetic nervous system branch. So in a state of homeostasis, we have both parts of the autonomic nervous system running in balance with each other. We have the sympathetic nervous system and the parasympathetic nervous system. And when the body is at rest, the parasympathetic is in most of the control. And this branch is known as the constant brake on the system. It's responsible for rest and digest functions. And as stressors occur, the parasympathetics will then mitigate their control of that brake, and the sympathetics, often referred to as the fight or flight system, increase its functioning. And the sympathetics are responsible for preparing our bodies for action by influencing our heart's contractility, dilating our pupils, and activating the production of catecholamines, such as epinephrine, or aka adrenaline. And when trying to understand the stress response, many studies look at the cortisol response, which is a very commonly studied stress hormone. But it operates on a relatively slow time scale compared to these beat-to-beat responses in the heart. So whereas these catecholamines, such as adrenaline, act really rapidly, and they often can produce effects on the mind and the body only seconds after secretion. So the stress response is often measured at acute and dynamic timescales by quantifying this rapid sympathetic drive, which allows researchers to then zoom in on these fine-tuned changes that are occurring moment to changing situations. And in this talk, I'll be referring to this overall process as allostasis, which is defined as the body's actions that it takes to adjust to its environmental challenges and to then return back to homeostasis. And additionally, older textbooks claim that it's purely that reflexive brainstem that's controlling the sympathetic nervous system. However, when you think about how your body can activate a sympathetic stress response hours, days, months, even years prior to a stressor actually occurring, it becomes clear to us that there's something more cortical happening with our nervous system. And now there's strong evidence from anatomic track tracing studies that the adrenal medulla, which is the organ responsible for producing these catecholamines like adrenaline, is innervated by the cortex. And the particular regions of interest seem to be the decision-making, emotive, and motor areas. So learning about the sympathetics allows us to truly understand a variety of interesting aspects of cognition, such as motivational states, stress reactivity, reward sensitivity, task engagement, decision-making, just to name a few. So prior to understanding how we measure the response of the sympathetics, let's first understand what even occurs in a heartbeat. So first, an electrical stimulation spreads across the upper atria, which causes them to contract and fill the lower ventricles with blood. And the electrical signal then moves down to the ventricles, causing them to strongly contract and pump blood out of the heart and through the body through the arteries. And this electrical stimulation and depolarization of the heart is seen with your classic ECG waveform, shown here in green in the upper right. And researchers often look at heart rate when studying stress. But the problem is that heart rate is quite generalized and it can be affected by many things, not just stress. It could be affected by anxiety and emotions, but it can also be affected by things like dehydration or body temperature or nutrition. And with both the parasympathetics and the sympathetics always at work to maintain that homeostasis, heart rate isn't very capable of zoning in solely at the sympathetic activity alone. So to look at these dynamic sympathetic activity responses, we need to be able to extract purely the sympathetic nervous system response. So the current most popular method that's used to non-invasively do this is by combining impedance cardiography, ICG, and electrocardiogram, ECG, to extract a temporal measure of pre-ejection period, or PEP. And PEP is a time estimate between the initial electrical stimulation of the heart, which is that Q point on the green ECG waveform, and the moment that the aorta actually opens to move the blood out of it, which is signified by the B point on the blue ICG wave. And it's reflective of the vigor with which the heart is contracting, which is known to be primarily controlled by the sympathetic nervous system. So a decrease in PEP signifies an increase in the vigor of the heart's contractility and an increase in sympathetic drive. And vice versa, an increase in the pre-ejection period time signifies a decrease in the vigor of the heart's contractility, which is then signifying a decrease in the sympathetic nervous system response. So with this variable, we were able to learn a lot about fast allostatic responses. And however, this method is generally really time intensive for researchers, and it has specific procedures that makes it difficult to regulate between test subjects in both application and analysis. But luckily, now we have a new bioimpedance method for measuring this activity of the sympathetics that simplifies both the data collection and analysis process, called Trans-radial bioimpedance velocimetry, or TREV. And the TREV method just uses these 4 strip electrodes placed on the arm, and it has the capacity to replace the need for ICG and ECG in estimating sympathetic activity. And TREV is capable of continuously measuring the impedance of blood flow through the arm by estimating the drop in resistance that results from red blood cells aligning in response to this pressure wave that flows through the arteries that's associated with that opening and closing of the aortic valve. So that's kind of a mouthful, but if you want to learn more about how this works and how we extract our waveforms from it, I recommend watching our previous TREV webinar where we dive into more detail on that. So here we have an example of 2 contractility heartbeats. And the amplitude of each beat here represents the contractility strength, which in turn represents sympathetic nervous system activation at that moment in time. So the greater the amplitude, the greater the contractility, that vigor of the heart's contractility, and the greater the sympathetic response, and vice versa. So the TREV method provides us with the ability to also estimate not only contractility, but also acceleration, velocity, integral of velocity, and cardiac output. But for this, we're going to be focusing on just the contractility metric in this webinar as our measure of the sympathetic nervous system. So to summarize, using impedance cardiography and ECG to analyze the sympathetic activity includes the application of 10 electrodes throughout the neck, chest, and torso, represented by the black, red, and white electrodes in the top diagram here. And with this combined method, researchers would then extract a contractility time estimate referred to as the pre-ejection period, or PEP, from the ECG and ICG waveforms. And while this method is effective, it can also be really time consuming and lead to some difficulty in analysis procedures, particularly with finding that specific B point notch of the ICG. And what's really convenient with TREV is that we can get that sympathetic nervous system measure simply from these 4 strips on the arm. And not only does TREV vastly decrease skin preparation and application time from something like 20 minutes for ICG and ECG to 2 minutes for TREV, but it also significantly cuts down on the analysis difficulty and time, in which you really simply just have to find those peak amplitudes of the contractility waveform rather than extracting a time measure between 2 points across 2 separate physiological signals. So that's my kind of summary of TREV in the past of sympathetics research. I'm going to briefly review a study which I ran in which I used TREV. So I ran a study which examined the interactions between the regulation of the sympathetic nervous system and cyclic fluctuations of sex hormones occurring in 30 healthy and naturally menstruating young adults across their menstrual cycles. So these are menstruating people that were not on any form of birth control and free of health abnormalities. And this graph here is an example of the fluctuating hormonal concentrations of estrogen and progesterone across a single natural menstrual cycle, in which, as you can see, sex hormones undergo drastic concentration changes. So for instance, as compared to menses, the menstruation time, there can be a 12-fold increase in concentrations of estrogen during ovulation and a nearly 80-fold increase in progesterone concentrations during the premenstrual luteal phase. And I brought these 30 participants into the lab for a total of 3 sessions each. So once per 3 distinct phases of their cycles, once during the menses, menstruation, once during ovulation confirmed by an ovulation test, and during the premenstrual mid-luteal phase, which is often associated with the PMS period. And the goal of the menses session here was to bring participants in during a time of low hormone concentrations across both estrogen and progesterone, while the ovulation session was scheduled to bring participants in during a time of peak estrogen, and the mid-luteal PMS phase served to bring people in during peak concentrations of progesterone and also high levels of estrogen at the same time. So I'll just quickly review the task structure and how we measured contractility. So participants were fitted with the TREV device and they completed a modular arithmetic task. And that's just a common task that's used by stress researchers to induce acute levels of stress by using arithmetic problems in a format that participants are unlikely to have come across previously in their lives. So the task consisted of a 2x2 design in which problems varied in difficulty from hard to easy and the amount of monetary reward, low versus high, that was possible to gain for each problem. And the overall task consisted of 248 trials of arithmetic problems. And throughout the task, the participants had to respond accurately to these problems before their trial time ran out. And the trial time was a challenging time limit that we personalized to each individual for each session. So here's an example of 3 consecutive trials of the task. And we're zooming in on the first trial. So first, a reward value of the upcoming problem is presented for 4 seconds. Then the problem is presented, to which the participant either responds to the task or the trial time runs out prior to receiving a response. And this is followed by a 4-second feedback screen, which informs the participant if their response was correct, wrong, or too slow. And for behavioral analysis, we looked at response times and accuracy on these trials, and we measured the effect on contractility, our estimate of sympathetics, in 3 different ways. We measured it in response to the trials, in response to the contractility following feedback, and as well as allostatic efficiency during feedback. So allostatic efficiency was measured by the rate at which contractility decreased following the feedback, aka like a feedback recovery. So for instance, once the feedback was presented, a slower rate of change from high to low contractility signified a low rating of allostatic efficiency, whereas a high rate of change signified a faster rate of contractility decrease, hence a quicker feedback recovery, representative of high allostatic efficiency. So for instance, in a situation of high allostatic efficiency, the body would increase the sympathetic drive when faced with a challenge, and then it would quickly return back to homeostasis by attenuating that system when that challenge has passed. And this is a summary of my progesterone findings. And as a reminder, progesterone peaks during the luteal phase, which is often associated with the premenstrual PMS phase of the cycle. And I found that progesterone was positively associated with response time and negatively associated with allostatic efficiency, specifically in response to difficult trials. So this suggests that higher levels of progesterone are linked to slower response times and a slower stress recovery on cognitively demanding tasks and vice versa. However, it is really important to note that this was irrespective of performance accuracy. So accuracy remained unaffected on all trials. But given that the main purpose of the allostatic system is to be adaptive and reactive, my primary theory for this progesterone effect is an adaptive one. So during the luteal phase, the body typically prepares itself for the possibility of a newly fertilized egg following ovulation. So for instance, the uterine lining will thicken to support a fertilized egg, whether or not that fertilized egg even exists. And then, as you know, the menses then releases that uterine lining. And I believe this preparation for pregnancy might extend into cognitive processes as well. So to reiterate, this link between progesterone and response time wasn't credible in response to easy problems or to performance accuracy. And that suggests that this isn't an all-inclusive general hindrance to cognitive ability, but rather an effect specifically attuned to tasks that require higher cognitive load. Additionally, our findings that progesterone is negatively associated with allostatic efficiency, I believe, may provide support for a threat detection theory during the luteal phase. So specifically, if progesterone is related to a heightened degree of caution and careful behavior to protect you in the possibility of pregnancy, then it's probably adaptive for that sympathetic response to remain heightened and vigilant following an environmental stressor, especially if that stressor can bear social repercussions like receiving feedback, which is what we were exactly looking at here. Lastly, here's a summary of my estrogen findings. And as a reminder, estrogen is the hormone which peaks during ovulation. And we found that estrogen was positively associated with the sympathetic activity in response to all the variations of modular arithmetic trials. And this effect persisted after receiving feedback as well. So our contractility results demonstrated increased sympathetic activity in response to all the parameters of the task and to performance feedback. And one theory here is that an increase in these dynamic bursts of sympathetic activity in response to environmental stimuli, it doesn't have to necessarily be this negative thing implying more threat-related stress. Instead, it could be a sign of excitement or a way of your nervous system to tell you that there's something meaningful and novel happening in your environment that requires your attention. And maybe it acts as a sort of pull from your nervous system towards interacting and engaging with your environment, which is what people have been shown to do more of during ovulation. And it's important to note that this increase in sympathetic engagement wasn't related to allostatic efficiency. So participants may be more reactive to stimuli, but this reactivity isn't hindering their allostatic recovery from the stressor. So that's a super brief and quick glance at some of my research into the sympathetics using TREV. And now we'll have Neil review some of the cognitive research that's been interested in the interactions between cognitive processes and the sympathetic nervous system. But first, I'll hand it back to Brenda for the poll.
Thanks, Victoria. Go ahead and launch this poll. And we're just interested in hearing more about your experience with SNS. And we'll just hang out here for a minute or so while people are responding. Seeing a little bit more responses coming in. It looks like it's kind of almost equally spread across the three. So yeah, about 35% of people are yes. Oh, we're going up a little bit now. 37% of people are yes, they are recording sympathetic nervous system. 26% of people are no, and 36% of people look like they'd like to in the future. All right, I'm going to go ahead and end that poll. Thanks everyone for participating. And now, Neil, I will make you presenter. Here we go.
Thanks, Brenda. Good morning, everybody. So to follow on from Vic's talk there, I'd like to just focus for a few minutes on some of the insights that our lab is getting into more complex cognitive processes and how we're using a combination of beat-to-beat physiological measures combined with neural measures to probe this issue. So for some time now, cognitive neurosciences has been revealing a lot of different cognitive mechanisms and the ways that cortical correlates can help us to flexibly adapt to the various challenges that we face in our lives. And in parallel to that, we've also been learning more about how responses in peripheral organ systems like cardiac reactivity is also co-varying with various different task demands, and especially tasks that require some kind of mental or physical exertion. A classic finding here would be an increase in cardiac reactivity indexed by lower PEP scores. The more you incentivize people to perform, in this case, a memory task or a cognitive task in general. But to Vic's point on the various different cortical pathways that can connect to sites like the adrenal medulla, our lab is interested in seeing if cardiac contractility as an index of the sympathetic nervous system is more fundamentally intertwined with more complex cognitive processes, given the ability for these different cortical sites involved in decision making and various other forms of executive control to communicate with peripheral organ systems. So for this, we've been using the approach avoidance paradigm, and the paradigm is essentially measuring a conflict that emerges when people have to face sort of the dueling appeal of an award and the revulsion of some kind of cost. So if I were to offer you a large amount of money in exchange for a tiny shock, chances are you would probably accept it if I offered it to you in a take it or leave it fashion. If conversely I offered you a large amount of electric shock and a tiny bit of money, again in a take it or leave it situation, the chances are that you would reject it, that you would not take it. So the interesting things happen then when the level of the money and the level of the shock come closer together. There's more parity between them. At this point, there's economically there's equal value to accepting it and rejecting it, but at a more cognitive level, you're likely going to be far less reflexive at this point. You're probably going to have to deliberate more on this decision. You're going to have to probably really hold the information in sharper focus and really compare the two dimensions of information that are presented to you. And at a computational level, you could think of this as more information bits or more evidence being incorporated into these more conflicting decisions. So what we wanted to do was to measure, as people are making these decisions, first at a computational level, what is the level of information that's being integrated into their choices. And to do this, we used a computational model that incorporates both the choices that are made and the response times. As you can see here, on any given offer, you will have a choice. So in this case, an easy accept trial will also have a response time associated with it. So in this case, the time it will take you to make that choice is going to be relatively small. Even if we were to increase the level of cost, you would, but not so much to the point that you would reject it. So in this case, you're accepting this offer as well, but now that increased cost is likely making you deliberate on it a little bit longer. Conversely, if we give you a pretty terrible offer here, you will reject it. It won't take you too long to make that decision. But again, we make the decision more conflicting. You may or may not accept it, and you're going to have a long deliberation time on it. Conversely, you might reject this offer and have a long decision time associated with it. So the important thing here to note is that the choices and the response times are relatively independent of each other. And so you can incorporate both of these into a model called the drift diffusion model. And one of the parameters that this model estimates from the patterns of choice and response time is how much information is being integrated into choices, with a parameter called the decision boundary. So it's important to note that this decision boundary is not something that we directly measure. This is something that we infer from the patterns of choices and reaction time. But the key prediction is that for these more conflicting choices, you're going to be dealing with, or that the model will suggest that there's a higher decision boundary governing these choices and a larger amount, or reflecting the larger amount of information that's being incorporated into the choice. On the other hand, if it's an easy accept or an easy reject, this decision boundary will likely return smaller, and in implying a more reflexive decision that doesn't require an awful lot of information to be processed. So a study we ran recently used this model with human data that was recorded, people making lots of these offers, varying the level of shock, varying the level of money, while we simultaneously recorded both EEG and impedance cardiography, electrocardiogram, in order for us to look at both neural correlates and correlates of cardiac reactivity, and in particular contractility reflecting changes in the sympathetic nervous system. What we were interested in seeing was when you put the sympathetic nervous system responses and neural responses into models of decision making that allow the decision boundary to correlate with the physiological signals, what comes out in terms of any correlations that are specific to the nervous, to the brain measures, or correlations that are specific to the sympathetic nervous system. Does it help having both? Do you just need the neural variables and so forth? And what we actually found was that you do in fact best explain the data when you incorporate both. We tested a whole range of different brain processes involving different frequency bands, different sites in the brain. The brain or the neural measure that seemed to explain the data the best was this measure of alpha symmetry. So what this means is, as you attend to the left and the right side of space, you will see increases and decreases in alpha power in the contralateral region of the brain. So here, for example, in the simplified schematic, if you imagine that reward is communicated by the size of the yellow bar and the shock is communicated by the size of the blue bar, you will see increases and decreases in alpha power recorded contralaterally at posterior sides of the brain. That will vary with the level of attention being given to each of these dimensions of information. What you can then do is compute a symmetry trace. So by that, it's the similarity in attention being applied to each side of space as an index of the higher this symmetry is, the more people are holding the information in a kind of a juxtaposition, or they're scrutinizing both in parallel, indicating that they're really deliberating over their decision. And the best fitting model also contained measures of the sympathetic nervous system. So we measured PEP. Here I've reverse scored PEP so that a higher score on the y-axis means a higher drive in the sympathetic nervous system. And what the model suggested was that there was an interaction between the neural and the peripheral signal. And the interaction looked like this. So if you imagine a situation where the alpha symmetry is low and the sympathetic nervous system is also not particularly active, you get a certain decision boundary. So again, the decision boundary, a higher value here, is reflective of the computational model thinking people are incorporating more information into their choices. You then increase the sympathetic, when the sympathetic nervous system was increased, we saw that the boundary was also increasing, indicating that the more people are incorporating information, there's more activation in the sympathetic nervous system. Similarly, there's an increase when there's a higher degree of alpha symmetry, or the more people are holding both the reward and the cost, they're attending to both channels of information at times when they have a larger decision boundary. But actually, the largest decision boundary was associated with both. So when you have a combination of the alpha symmetry and the attention to both channels of information and drive in the sympathetic nervous system, those are the moments when people are in our data set, people had the largest decision boundary, indicating the maximum time of a conflicting decision that they were really incorporating information into. So to delve a little deeper into this and to see, again, if there's a fundamental relationship between the sympathetic nervous system and some aspect of this cognitive process, we took a look at a slightly different way of looking at the brain data, which is to look at phase coherence. Think of this as, rather than looking at the size of the waves, we're now looking at the temporal consistency in when the waves emerge. And what we find is when the sympathetic nervous system is high, we see that there's a dramatic decrease in the phase coherence in alpha, but specifically related to the reward information. We don't see this decrease or any kind of change in the phase coherence of alpha reflected to the shock information that responded to the level of the sympathetic system. So we concluded, or we interpret this data potentially as meaning that as humans, we tend to like good information, we tend to like good news, we maybe have a bias towards looking at reward and rewarding things. When it comes to a more conflicting decision where we need to really scrutinize all available information, including things that we may not want to look at, like things like cost, there is potentially a requirement for the sympathetic nervous system to be active to support a scrutiny of multiple channels of information, particularly when we need to maybe disrupt processing of a prepotent signal like our bias toward reward. On a broader level, our work is showing the value of integrating computational modeling, neural data, and measures of the sympathetic system in parallel, particularly when you can look at the short time scales that allow you to look at different choices and moment-to-moment changes in these signals. And more on the theoretical level, our work is demonstrating that there is quite likely more to sympathetic reactivity than mobilization and exertion, and it might have a more fundamental role in intertwined in complex processes that help our behavior. And with that, I will hand back to Brenda for the next poll.
Great, thanks so much, Neil. Let me go ahead and launch this poll. And now we're interested in learning about your use of impedance cardiography. So we'll give people a minute or so to respond to this poll. And then if you do have questions for our speakers today, you can ask them at the end. We won't have a ton of time for questions today because we have a lot of content that we're presenting, but we're certainly happy to answer a few. Okay, so let's take a look here. It looks like we have about 10% who are using impedance cardiography, 48 are not, and 41% are looking to do so in the future. Thank you so much for participating. Now we will switch gears to Elizabeth. I'm going to go ahead and make you presenter, Elizabeth.
All right. Hi, everyone. As Brenda mentioned, I have a position at UCSB where I act as a liaison between UCSB researchers and BIOPAC to facilitate research using BIOPAC products. And as you saw in Neil's presentation, labs in the department, including ours, have typically used impedance cardiography in the past to look at sympathetic nervous system activity. Recently, there's been growing interest in TREV across different research areas, such as social psychology, developmental and evolutionary psychology, and cognitive neuroscience. To my knowledge, there are at least 6 labs in our department who have utilized TREV for their research. And researchers pair TREV with a variety of techniques to measure relationships between the body, brain, and behavior, including the respiration belt, ECG, EDA, brain functional measurements such as EEG and MRI, and computer tasks that might assess motor or cognitive skills. And researchers are interested in measuring sympathetic nervous system activity correlated with or in response to emotional or stressful stimuli, brain function signals, endogenous biological factors, and or cognitive performance. So some current questions that researchers at UCSB are investigating with TREV include, do sympathetics respond to a more versus less stressful environment during spatial navigation and predict performance? Do sympathetics synchronize with brain function measured with EEG and visual attention performance before versus during a stressful stimulus? Do sympathetics vary with hormonal fluctuations across the menstrual cycle or across the diurnal testosterone cycle? And a question that my lab is currently working on is, do sympathetics synchronize with brain function as measured with MRI in preparation to move to win reward, prevent loss, or avoid punishment? So our lab used to do experiments with combined impedance cardiography and ECG, as you saw in Neil's presentation, in order to get the pre-ejection period, which indexes sympathetic drive. Since then, our lab has transitioned more away from using ICG to using TREV to get contractility measures that also index sympathetic drive. And the main reasons for making the switch is that the setup is much quicker and easier, as Victoria mentioned, and that locating the ICG-B point is often very difficult in ICG MRI data due to MRI artifact. So for impedance cardiography paired with ECG in the MRI, you have a process of placing many different electrodes in a special configuration in order to minimize MRI artifact effects. But TREV, on the other hand, is a simple process of applying 4 strip electrodes and body tape to the arm after the participant has washed their arm with soap and water and patted it dry. And since TREV is placed on the arm, it benefits from not being near the chest and head area that's in the middle of the MRI bore. And TREV can also be paired with other devices in the MRI environment, such as EDA. However, for both ICG and TREV, the MRI environment can raise significant safety concerns. Because the machine is a large magnet, essentially, there's potential for electrodes or leads to burn the participant due to the creation of an electromagnetic loop. The ICG setup required that a pillowcase be placed as a barrier between leads and participant skin, and that the leads would be as straight as possible in order to avoid loops. TREV is similar in that we don't need to do this complicated setup, thankfully, but TREV is still capable of generating loops. The TREV strip electrodes can wrap around the entire wrist of a participant in those with smaller wrists and overlap onto themselves. So before applying the electrodes, we will measure a participant's wrist with measuring tape. If a participant's wrist circumference is smaller than the length of the TREV strip electrode, we will cut a little bit off the edges of the electrode so that there is space between the ends so that we break the loop. And we try to cut as little of the electrode as possible while still allowing for that space. One limitation of TREV is that it's very sensitive to arm muscle tension and movement, so it may not be the best option for studies that require use of both hands and arms during the experiment. But one benefit that we found in our studies of the MR environment is that the scanner actually can restrict arm movement for the participant due to their positioning in the scanner. And for our studies, we've actually been able to collect a lot of data with relatively little movement noise, even though participants are completing a joystick task with the other arm at the same time. Though TREV is easier in many ways, the MR environment can still significantly affect the quality of the signal, as with other psychophysiological measures. So most of the time, in most of our participants, the TREV contractility wave looks pretty clean before the MRI sequence begins. And usually the better it looks during the sequence, the more robust it will be to MRI noise. The better it looks before the sequence, the more robust it will be during the scanning sequence. So it's in your best interest to test the signal and do some troubleshooting before you begin the scanning sequence. But once the sequence begins, it can look like this. TREV is capable of being quite robust to MRI noise, and you can see here that the peaks are clearly defined and that the noise causes a little bit of jitter in between peaks, but this isn't a problem, and this data shown here would be relatively easy to analyze post-collection. On the other hand, if something went wrong, it can look like this. And here you see that the contractility peaks are lost in the MR artifact and that the peaks are quite distorted. So how do we prevent this from occurring in the first place? The ability to get a clean signal in the MRI depends on a variety of factors. Both in and out of the MRI, the TREV signal quality can decrease due to improper skin cleaning. We actually recommend against using alcohol wipes to prep the skin for TREV because they can dry the skin out and can reduce signal quality. Imprecise electrode placement can also negatively affect signal quality, and other participant factors such as individual differences in arm circumference, participant motion, and muscle tension can also affect the signal. In the MRI, different scan sequences such as a T1 MP-RAGE anatomical scan, an echoplanar imaging scan for functional MRI, or a diffusion scan for structural diffusion measures can cause varying signal effects. And scan parameters such as the multiband factor, TR, and whether you're doing a single versus multi-echo scan can vary in how much noise they introduce into the data. So when you're piloting, you might want to try out different sequences with TREV and other psychophysiological recording devices in order to see which combinations are at risk for higher noise. In our data, we identified that the frequency of the MR gradient flip, which can be calculated from the multiband factor and TR, was distorting our signal. So we've explored different solutions for processing our data, and which option is best will vary from study to study. For relatively clean data with a few distorted peaks here and there, we've played with a smoothing procedure where we replace individual compromised peaks with an average of surrounding peaks. And for messier data, we've explored filtering out the MR gradient noise because we were able to identify the specific frequency for our studies that the scanner was introducing. But if you're going to look into doing this, the frequency of the MR noise will vary based on which scan you're working with and can vary based on all the factors that I just listed. So you should proceed with caution with this method to be careful to not mess with frequencies that contain real physiological data. Now I'll give you a little preview of our recent work that involves collecting TREV and MRI data. And one task that we have been giving to participants in the scanner is called the active escape task. And the purpose of this task is to assess the function of a brain motor network that is potentially jump-started by an emotionally arousing experience. And this task was designed and collected by Joanne Stasiak, who's another PhD candidate in the department. How the task works is that participants are presented with a cue that informs them of which type of trial they're about to complete. Then they're presented with an 18-second countdown where they must wait until the trial starts. Once the trial begins, they're tasked with moving a cursor, which is the triangle, with a joystick to a goal space, which is the circle, and they must move this cursor as quickly and accurately within a specific amount of time. Once they do so, they're asked about the intensity of their emotional experience on a scale from 1 to 4. And you can see that the trial types are drawn from a 2x2 design. Trials are either controllable, which means that you can avoid the shock punishment by completing the motor task quickly and accurately, or they are uncontrollable, which means that the shock will happen to you regardless of your performance. And mild versus unpleasant trials tell you the strength of the shock that you will experience. And some exciting preliminary data that Joanne has recently found is that contractility during the countdown period, so that 18-second countdown before they must move to avoid a shock punishment, was significantly positively correlated with subjective ratings of emotional intensity. So the higher a participant's contractility is during that countdown period, the higher they will rate the trial as an emotionally intense experience when they're asked on that scale from 1 to 4. So thank you all for your attention. I want to highlight our lab's recent paper where we tested TREV with a grip strength task, and it's published in Psychophysiology. And you can find a lot of great information here. And if you use TREV, you can also cite this paper to show that TREV has been previously tested by researchers. So now I'll hand it back to Brenda, and after that, Fraser will be discussing the equipment and setup required to collect TREV data in the MRI.
Great, thanks, Elizabeth. I'm going to go ahead and launch this poll. And where do you record your data? Let us know. Oh, let's see here. It looks like, okay, now it's launched. Here we go. Let's see where you're recording your data. Looks like In the Lab is winning. Most popular. Few people doing MRI. A few people doing in the lab, in the field. And let's just give it another 10 seconds or so. Okay, yeah, it looks like 64% in the lab, 24% MRI. No one is doing both. And then in the field is about 32%. So thanks all for participating in that. And now we are moving on to Fraser Finlay. Fraser, I'm making you presenter.
Thanks, Brenda. All right, thank you, Victoria, Neil, and Elizabeth for those great presentations. Lots of really useful information there. I'm going to take a look at the hardware options we have for using TREV for the different use cases that have been discussed. So to start with, I'm going to look at the MRI application use case. I'll start with this option because there's some unique considerations that are specific to that particular environment. The MP160 is the only platform we offer that is suitable for MRI. And BIOPAC has developed an entire line of products that are specific to recording physiological data inside the scanner. The MP160 is a 16-channel system that records physiological signals, and then you can also use it to deliver electrical stimulation, and that can occur inside the scanner as well, and then synchronization with visual presentation systems. Now, one of the keys to operating in the MRI is to try and get the cleanest signals possible. And to help that, BIOPAC has developed MRI smart amplifiers. And these incorporate advanced signal processing circuitry, which removes spurious artifact from the source physiological data. Signal processors inside the amplifiers are able to distinguish between the physiological signal and the MRI artifact as manifested by gradient switching during MRI sequences such as shim or EPI, for example. Because MR-related transient artifact is removed at the source, this means that you can treat this amplifier the same way you would treat any of our other amplifiers with regards to recording the signal at a typical sample rate, so no additional considerations outside the use of the MRI specific amplifiers at this point. In addition to the amplifiers, we offer a whole range of transducer options, both electrical and pneumatic. So if you're adding in grip force, for example, that can be done with TREV, respiration, a whole slew of different items. If there's anything that you're not seeing on the list, let us know. The chances are we can do it as well. In terms of the electrodes that we use are carbon fiber, and they're safe inside the scanner. Same goes for the electrode leads. The lead 108 series is the primary electrode for a lot of the biopotential type signals. These are snap fit and they connect to our different electrodes If you're using the EL526, which is specifically intended for TREV, those electrodes have the leads connected to them, and they'll connect directly to the patient cable, which I'll show in a moment. And as Liz mentioned, you can cut these down to fit. You want to avoid a situation where you're creating a continuous band around the circumference of the forearm. If you're collecting other signals simultaneous with the TREV signals, we have the EL509, which is a dry electrode for electrodermal or intended for electrodermal activity. When I use the term dry, this electrode does require gel, but when you peel it off the tray to apply it to the participant, you do have to add the EDA specific gel. If you're recording ECG and EMG, the circular electrode underneath is the EL508. That is a pre-gelled electrode. These are all carbon fiber, super safe inside the MR. Just what you need to do is make sure that you've got really low impedances with these types of electrodes, because the higher the impedance, the more potential you've got for gradient noise coming through. We have the L check, which is a very straightforward impedance checker. You connect the electrode leads and the electrodes to the device, and it will measure the impedance and give you information concerning poor contact between the electrode and the participant. Okay, so again, the MRI area, we break it into two rooms. The room where the BIOPAC equipment is located is typically the control room, but that may not always be the case. The MP160 system, any of our amplifiers, they remain outside of the chamber room. The participant is obviously in the chamber room. The electrodes, the electrode leads, the transducers that are appropriate for MR use, those are all suitable for the chamber. And then we have a series of cables. If it's electrical signals or if it's pneumatic, we go through the waveguide. The cables connect to the penetration panel. A filter is mounted on the panel, and then the cables pass via that particular filter. So on the chamber side, we have a long cable, patient cable. It's 8 meters in length. One end, the electrode leads will connect into. The other end connects to a filter, which must be securely mounted to the penetration panel. This is probably the #1 culprit for a lot of unwanted noise that comes in. Make sure that this device is securely fastened. It comes with a bracket. The filter has a connector on either side of it. One side is intended for connection to the patient cable on the inside of the chamber. The other side is intended for connection to the amplifier cable in the control room. And the black end of that cable connects to the amplifier. We have a variety of those cable sets. They're specifically designed for MR, and they're specifically designed for different signal types. So check with us and we can give you advice on the correct cable for the signals that you're recording. If you go onto our website, we identify things that are MR applicable, and we break them into two categories. The first one is MR conditional, and whenever we describe something as MR conditional, we define the conditions of use. So in this instance, this device is limited to a 3 Tesla field strength, but it could be something else. Then it may need an additional cable that has to go with it. In any case, the conditions of use are always defined. if we define something as MR safe, that means that you have no additional considerations to account for. In this case, this is our pneumatic respiration amplifier, so that's fully safe. The pneumatic line then passes through the waveguide to the amplifier in the control room. Okay, so for those of you, and it seemed like more of you are doing non-MR than MR, we've got quite a few different choices for you. The top right image shows the D-series smart amplifiers. These are really nice. They're plug and play, very small. You plug it into an AMI 100D and the software identifies the amplifier, and it uses the same electrode lead sets as our wireless biopneumatics module shown below. The taller rectangular box with the antenna on it is the receiver unit. That attaches to the MP160. And then in the foreground, the smaller unit, that is the transmitter. So the transmitter is mounted on the participant, and then the signals are transmitted back to the receiver. And you can have multiple devices. These are quite often dual channel, and you can have any combination that we offer. So that gives you a tethered and a wireless option. Those are our sort of current platforms. If you're using the EL526 with either of those devices, you will need the cable shown down below. That's the CBL246. One end plugs into the amplifier or the transmitter, and then the electrode leads will plug into the mating connectors. So it's easy to set those up. Some of you may already have our impedance amplifiers. We've got the NICO 100C and the EBI 100C. The NICO 100C is more typical for impedance cardiography and is what we typically recommend for TREV. These two devices interface with an MEC 110C that connects to the front of the NICO or the EBI. And then we use lead 110 electrode leads to connect to the amplifier if you're using the transthoracic method. Or if you're using TREV, then you connect the 526 electrodes directly to the patient cable. So pretty easy to set that up. We've got lots of different solutions for measuring sympathetic nervous activity. These devices are going to be specific to your particular use case, whether that's in lab, MR, or remote. If you've got questions about any of these, reach out to us. Our sales and support team can definitely give you recommendations and advice on best practices for setting the participant up and what combinations work together. Before I wrap up, I do also want to point out that the term trans-radial electrical bioimpedance velocimetry, TREV, was invented by the late Dr. Donald Bernstein, and there are patents and references covering this technique as well, which you can look up. And then finally, from me, BioPAC hosts workshops around the world. These focus on, either different products or different recording techniques and application areas. If you're interested in attending a workshop in your area or even hosting a workshop at your facility, you can reach out to us and we'll provide you with further information. Well, that concludes my part of the presentation. Thank you for your attention. I'm going to hand back to Brenda.
Thanks, Fraser. I know we are over time here, so I am going to go ahead and share my screen, and I want to give you a couple, some information about a couple more things here. I am looking at my screen, okay, here we go. So first, I just want to show that Victoria mentioned our other webinar, and it's the impedance and cardiac output with TREV webinar, where it goes into a lot more detail about the TREV process. And you can access that on our webinars page, which is biopac.com webinars. We actually have over 100 webinars here and lots of great information for you depending on the topic that you're interested in, but TREV is definitely there, so you can access that easily. Also, I want to let you know that on the bottom of any page of our website, if you want to reach out to your local sales rep and ask a question, you can just click this here and fill out this form, and an email will go directly to them wherever they are in the world, and they'll reach out to you, usually within 24 hours. And we did have one question. So let me just go ahead and share my screen here. Sorry, increase my display of the slides. Okay, so we had one question from Gary, and let's see, let me just open this up. Okay, I'm curious if TREV could serve as a proxy for gauging a system-wide sympathetic response instead of analyzing heart rate variability through power spectral analysis, such as overall sympathetic nervous system induced thermogenesis. And Victoria, I think that one's for you.
Yeah. Great question. So it is different doing SNS research in the ways that we've discussed today from analyzing heart rate variability and power spectral density, in the way that contractility of the heart is known to be directly controlled by the sympathetic nervous system. So it's not just taking heart rate and trying to extract and pull apart parasympathetic, sympathetic, and estimate it as a sympathetic control of heart rate. It is a direct control of the contractility. So by that, estimating that contractility is a estimate of the general activity of the sympathetics, either increasing or decreasing their actions. And, all the way from the catecholamine response to the systemic changes of the sympathetics. So I would say that it is different from the heart rate power spectral density analysis in that way, that you could apply it in a more systemic way of what are the sympathetics doing that are then causing this contractility difference in the heart as well, either increasing or decreasing its function. So hopefully that answers the question.
Okay, well, thank you very much. I think we are over time, so let's move on. I already mentioned that we have live webinars almost every month of the year. Our next webinar is scheduled for May 23rd. It's on electrical stimulation. So check back at our website and make sure you're signed up to receive our emails. And you're more than welcome to join us live for those events or watch any of our webinars on demand. Just some closing comments here. Today's webinar was recorded. I'll email you a link with today's recording and slides if we're allowed to share those. When you close the webinar window, if you see a survey, please complete it so we have your feedback and ideas for future webinars. You may visit biopac.com for additional references, including screencasts on specific features and analysis tools, application notes with step-by-step instructions, and information about future events. We are live at events, so find us and come see us and chat with your regional rep if they're there. Speakers, anything else before we conclude? All right.
I think we're all good.
Thank you very much. This concludes today's webinar. Have a great rest of your day, everyone. Take care. Bye.
Thank you. Bye, everyone.
Bye.